So most of the current, basically, regenerative medicine and tissue engineering research relies on the use of animal tissues or animal biomaterials to make those bioinks. So if we take, for example, like in the case of collagen, if we are going to use a bioink made out of collagen, we can use rat tail collagen, we can use bovine collagen, we can use porcine collagen, and probably fish collagen. In bovine collagen, we have collagen from bone, we have collagen from skin, and so on. So we have all these different options. So aside from the fact that the manufacturing technique changes the properties, the source tissue is actually contributes to a lot of different properties that we've been seeing with these different basic bioinks. We know that at the end of the day, that rat tail collagen is suboptimal to the human skin collagen. It has been documented well in the literature with the idea that there are differences of amino acids, and even minor differences in amino acids contributes to major differences in cellular response. Now we've been talking about the Modernization Act, the FDA Modernization Act of reducing or eliminating animal testing with the option of having something as good as a human model or organoids to basically predict the toxicity level of drugs, so we do not basically get to spend a lot of time and money into irrelevant animal testing before transitioning to human studies. So basically, as a company, we realized that if we want to advance the field of regenerative medicine with clinically relevant therapies, we have to introduce human-derived biomaterials that they are actually compatible with the cells, and they basically have a better translational aspects compared to other biomaterials. So as a company, we provide high quality, affordable, and stable supply for human derived biomaterials. We started with three main products: the human collagen, because it actually compromises more than 90% of our human tissues and organs; human gelatin, we heard from Anis and many different speakers today about the use of gelatin and ferritin in the morning; and also a human extracellular matrix that can be used in a similar fashion to Medrogel. So our unique platform actually works on the idea that we support the idea of limiting the use of animal in testing and animal-derived biomaterials. As a company, we work with partners to support their FDA submission, so they can actually utilize these biomaterials in human therapies. We work only with tissue banks, so basically we save them the cost of discarding that high-quality tissue. And as a company, we're actually FDA registered and inspected facility, and we are compliant with medical device and tissue practices. From these